Hey everyone, Mr. Murphy here. I'm going to go over chapter 5.4, which is all about the movement of larger molecules across the cell membrane. So macromolecules are typically too large unless they're broken down into their monomer form. And so there's also circumstances where not just macromolecules, but maybe like bacteria cells or other things need to be taken into an animal cell or a single-celled organism in order to be broken down, or any number of things. The point is that those are far too large to simply pass through a little protein. If you had a protein large enough to pass bacteria cells through it, you're probably letting everything in, right? Because it's too big. And so the ability to secrete or take in these molecules is referred to as endocytosis or exocytosis. Endo meaning going into the cell, and exo meaning coming out of the cell. So here's an example of endocytosis. We have a very large macromolecule or some sort of large body here, and that's going to create an indentation in the cell membrane. That's going to be mediated by a whole bunch of different proteins. So don't think that it's just like, oh, it bumps into it and it just enters. It's a much more complicated process that probably involves different receptors being here, which then will attract other proteins from inside of the cell to mediate the bending and folding of the cell membrane. All of that's way more detail than we need. I just want you to understand this is not like a spontaneous thing. This is very much an active process, which is going to be requiring ATP or some form of energy carrier in order to happen. But basically what happens is that as that molecule is taken in and you get this indentation here, it's eventually going to be pinched off, again with the help of different proteins that'll facilitate the breaking of the membrane here, which will repair the external membrane while allowing this little vesicle now to exist inside of the cell. Recall that we talked about how there's a recycling of membrane material, that these phospholipids, they don't exist in a single membrane for their entire lifespan. Not that it's a lifespan, but their entire existence isn't necessarily spent on one membrane surface. They may become parts of a vesicle, they may fuse with a lysosome, ultimately end up as part of the endoplasmic reticulum or even the nuclear envelope around the nucleus. So these individual phospholipids get tossed around as needed. So this is a little endocytotic vesicle. Maybe that joins up with a lysosome and ends up getting broken down, or any number of things could happen depending on the specific example. Exocytosis is exactly the opposite. Maybe we've just made a whole bunch of insulin or digestive enzymes or things like that, and we need to get that out of the cell. That vesicle will bind to the cell membrane, again, with the help of proteins. That will facilitate the joining of these membranes and then the release of the materials. And pay attention, the membrane material right here unfolds and just becomes part of the cell membrane. So that material it's not used up, it's not lost, it's just a constant recycling of this phospholipid material between different sources. So there's three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. In all three, the membrane is indenting in. Invagination is the term for it. It's uncomfortable. Which forms around the molecule and creates that vesicle. Phagocytosis, which is like cell eating, is how you should think about it, is when we engulf a large particle into it. Food vacuoles like phagosomes form, which then fuses with a lysosome, which has all the enzymes, which then breaks it down and turns it into monomers, and then those monomers can be released into the cell wherever they're particularly needed. So that's a way in which cells actually will digest the things that are brought into the given cell. We see this a lot with single-celled organisms. It does happen within animal cells. However, often much of the actual digestion of like food, like real food, is occurring within like the lumen of your intestines and things like that. So it wouldn't be like, chunks of a cheeseburger are actually making their way into a cell and then being broken down. Our bodies, we've advanced beyond that, and we let that be handled in the intestines. Pinocytosis, very similar. Cell drinking, so think of this as a way in which large fluid intake can occur across the membrane, and this is a very similar process. It's just a matter of like what is actually being taken into the cell. And the reason it exists is just because there's so many different circumstances where different cells need to take in different things. Yes, on a detailed level, this would be very different in terms of how it's controlled and all of that, but ultimately it looks the same as phagocytosis in terms of like something is being brought into the cell. Receptor-mediated endocytosis, so I had mentioned when I showed the diagram of the forming vesicle that receptors might be involved in this, and these would be proteins on the extracellular surface of the membrane, the exoplasmic side, that would then have a particular shape and grab onto a particular ligand that then would facilitate the movement of a molecule into the cell. An example of this is clathrin, which basically what happens is you have these particular receptor proteins, something on the outside of the cell, a ligand of some sort binds to these, which then encourages the creation of this vesicle, and we call it a coated pit here, that these receptors have found their ligands, and they are therefore leading to these clathrin molecules. Through a series of changing shapes of proteins will lead to these clathrin molecules to facilitate the bending of the membrane inwards, which then facilitates this. Basically, this is like one step closer to a more realistic example, where just demonstrating that none of this is happening super simply. It's all very well controlled and mediated. There's a reason why we point out that there are over 10,000 proteins in any given cell, like 10,000 different types of unique proteins inside of a cell, because the cell is super complicated. So when I say that, like, oh yeah, to make a vesicle you need like 18 different proteins, it shouldn't be that surprising, just because there's so many different proteins serving very specific roles inside of the cell. And what's kind of cool about this diagram is you can see that it is modeled after a actual microscopic examination of this process in action inside of a cell. Exocytosis, reverse the whole thing, just send it back out instead of in, and that's important for the secretion of waste products. It's important for the secretion of things made by the cell that need to be delivered elsewhere within the organism, and that's how we move large things. If you have questions, let me know. This would be another section, kind of like the cytoskeleton in the last one, where as long as you understand the broad strokes of what's going on, you're in good shape. Thanks.